This is a model of a signal transduction pathway using epinephrine as a signal hormone in a liver cell that will ultimately lead to a cascade of reactions, and finally glycogen will be broken down into glucose, which can be used as an immediate energy source by the body. So to start the reaction, epinephrine, the signal ligand hormone, will bind to the G protein receptor, which is located in the cell membrane of a liver cell. This reaction will convert this energy carrier molecule GDP into GTP, giving energy to the G protein that's attached here to release its alpha subunit. So its alpha subunit is released and will then move down the cell to an inactive protein, an enzyme called adenylyl cyclase. However, once the subunit, alpha subunit, binds to adenylyl cyclase becomes activated. And the job of adenylyl cyclase is to convert ATP, another energy carrier molecule, into cyclic AMP. So again, that's a conversion of ATP into cyclic AMP. So it's gone from a triphosphate to a monophosphate molecule here, and cyclic AMP is the secondary messenger in this cascade reaction. And so actually the binding of a single epinephrine hormone will lead to a reaction where hundreds of these cyclic AMP molecules are produced. What's their job? Well, cyclic AMP in this cascade reaction will then bind to protein kinase. And there are two parts of protein kinase in my model here. This part right here is the regulatory part. So this inactivates the kinase. However, when cyclic AMP binds to the regulatory part of the protein kinase, it actually removes that part. So now the catalytic part is free to react. And it will actually interact with, imagine there's another A here, it will actually interact with another ATP molecule and will receive a phosphorylation. So it's going to pick up those phosphate groups. And now we say that the protein kinase is activated. And so the protein kinase, its job is simply to do a phosphorylation reaction. So it's going to catalyze a phosphorylation reaction where it's going to transfer its phosphate groups to this enzyme phosphorylase. And the job of phosphorylase is then to, now it's activated, now that it's been phosphorylated, it will come over here to glycogen, which is stored glucose. So it's stored glucose linked up in a chain, and it will pop those bonds. So it's going to come along and it'll actually break these bonds here and start to release glucose. And so these are individual subunits of glucose that can then leave the liver cell, can exit the body here and go into the bloodstream, and through this glucose transporter leave the cell and then can be used as energy for the fight or flight response.